talk about Geneshot, and then I will have a brief demo of the website. So obviously all of the IDG project is kind of evolving around this idea that there is a lot of understudied genes, and on the other end, you have a lot of overstudied genes. So we can see this in data that we have from user behavior, for example, from this website Enricher, where we have millions of user-submitted gene sets. And we can see, if we just look at the frequency of the genes that are submitted to this website, the top most cited genes that are highlighted in the left are also the genes that are most commonly uploaded to analysis websites. And by just breaking down these frequencies to lists that have very few genes and a lot of genes, we see that the shorter gene lists are more heavily biased towards overstudied genes, meaning as soon as there's some manual input, we get these biases. And so this really led to the idea that we should maybe try to predict genes that are understudied and associate them with gene functions. And so we did this in the ArchES4 project, where we kind of just used the average correlation of a gene to a gene set, given some gene-gene similarity matrix, and that works really well. So for this, we just tried to predict the biological processes, pathways, upstream kinases, and transcription factors. And over the board, just using the ArchES4 data and the simple average correlation score, we get like fairly good predictability of gene function. So to make this more accessible, we built this web tool called Geneshot. And Geneshot tries to combine literature search with this function prediction that we started playing around with in ArchES4. And so in Geneshot, in order to find similarities between a gene and a gene set, it uses different types of similarity matrices that you can choose from. So you either use gene-gene co-expression information from ArchES4 or literature co-occurrence. So if two genes co-occur in a lot of publications, they will be likely functionally similar. And then we can also have a similarity matrix based on the Enricher queries co-occurrence. Yeah. So then the advantages of just using this simple metric is that we don't really have to deal with overfitting, because it's just calculating average correlation. And we have a very high coverage because we're using this high throughput data in case of co-expression. So all the understudied genes are just as well represented as overstudied genes. So just in basic terms, how this works is we perform a PubMed query that is composed of biological terms of interest. So for example, you could search for publications that mention liver, but not cancer, for example. So then we get a list of PubMed IDs. This gets cross-referenced with two databases that we have that contain publication to gene mappings. And so that will retrieve us a list of genes that match our search term. And this set of genes we can then augment with the gene-gene similarity matrices in Geneshot. And this gives us novel potential genes that are associated to the publications that we searched for, but are potentially not mentioned in that context before. And then on top of that, we also implemented this gene function prediction that allows us to associate different gene set libraries and their functions to an individual gene. So in this example, this is TNF, and you can see as the result page loads, we look for KEGG pathways, and so the top 3 hits are already known, so they're marked in blue, but then the 4th and 5th are novel hits, so potentially this gene might be involved in those terms, and you get like an AUC curve. And then also, if you have your own gene set, you can just upload that, and it will find genes that are related to that gene set. So let me go, and then I can do a quick demo. So here we have basically a website. So it's when you load it, it's just this input mask, and you have a search term for terms that you want in your query. And you can then also add terms that you do not want to be in your query. So you can filter out publications from certain contexts. So if we just load something like this, so this takes a little bit of time because we basically relay that query to PubMed, and that query sometimes takes a little bit of time.
I don't think we could see the demo.
Oh, sorry. Yeah, let me. Sorry, that was not so smart. There we go. Okay, so you should be able to see it, right?
Yeah.
Okay. So I ran this query, and so now what we get is this scatter plot down here. And so each point represents a gene. And on the x-axis, we have the number of publications that matched the search term and also intersect with this gene. So there were 2,400 samples that matched glioblastoma and also MGMT. And on the other end, we have the frequency of like the total number of mentions of the gene and the number of papers that were actually about glioblastoma. So MGMT seems to be pretty specific for the search. So then if we scroll down a little bit here, we come to the prediction side. Just a second. There we go. So here we come to the prediction section. So the table on the left is basically just a representation of the scatter plot that we saw before. So here we have our top genes, and this is by the frequency. And here we have our predicted genes. So these genes were calculated to be similar to the genes that we found through the literature search. And they were related through arches for co-expression. So here you can also select different ones. So we have these are the literature co-occurrences, and here we have enricher co-occurrences. But there could potentially be other similarity matrices in the future. We can also filter by understudied genes. So for example, a dark kinase, this is like an understudied kinase that seems to be related to the search term. So this could be an interesting lead. And you can submit these to enricher as well. So then the gene function prediction that I've briefly shown. So this is the example that I just showed in the slide. So this basically just runs the prediction, and these are the top pathways for this gene. And you know, instead of KEGG pathways, we have many other categories. You can also look for mammalian phenotypes, which is based on gene knockdowns and observed phenotypes, biological processes, and so on. And then the gene set augmentation is where you can just paste in your list of genes that you might have found maybe from differential gene expression or some other study. And so here we have 3 examples where let's click on mixed genes. So these are kind of genes that are mixed in terms of their rarity or commonality, like how common they are in the literature. So this is somewhat mixed. So there's 5 very commonly cited genes, and then 1 rare gene and some uncommon genes. So this is the list that we uploaded. And then here we get the list of genes that are associated to our input list. And again, so here you can change the similarity matrix from which this list is calculated. And by changing here, so this is a manually curated publication to gene similarity, and this is an automated one that's using NLP to get these the statistic, but this doesn't influence the prediction. So yeah, on top of that, we have an API that can be used. So everything of these things is calculated by using microservices. So they're all accessible programmatically. So you can calculate these similarities using a Python script or R. So this is just exposed to the outside. And yeah, so this is the quick rundown of the website. There is a help section if you have questions. We made a video about it. To read it, it explains more or less all the similarity matrices, how they're built. So yeah, that's it. I hope it's useful. And if you have questions, feel free to ask them. I'll try to answer them in the chat, I guess, for the sake of time.
Thank you.